Welcome to the Radiology Advances podcast.
Today, we're diving into a paper published in Radiology Advances titled Optimizing MRI Annotation Workflows for High-Accuracy Deep Learning Thigh Muscle Segmentation in Athletes.
The first author is Alexis Slutsky-Ganesh from the Emory Sports Performance and Research Center in the United States, with second author Salome Baup and senior author Augustine Ogier from the University of Lausanne in Switzerland.
The central question this paper tackles is a highly practical one. Exactly how much annotated ground truth data do you need to train a fully reliable deep learning segmentation model?
Right, and the clinical target here was the human thigh, specifically segmenting 14 thigh muscles on MRI in a cohort of professional football athletes and patients with acute ACL injuries.
But before we get into the algorithms, we need to establish the clinical stakes. If you are managing an elite athlete's recovery, why is it so critical to perfectly isolate, say, the vastus lateralis from the rectus femoris on a scan?
Because the clinical landscape has moved way beyond qualitative assessments. Just noting that a patient's quads appear generally atrophied following an ACL tear is not acceptable for elite sports medicine or rigorous research. We need quantitative MRI biomarkers, or qMRI.
Monitoring these specific numbers allows us to plot a highly accurate trajectory of a patient's recovery long before macroscopic changes are even visible to the naked eye.
Right, but extracting those numbers requires isolating the exact pixels that belong to the muscle and excluding absolutely everything else.
If your segmentation algorithm accidentally includes a pocket of intermuscular fat or clips the adjacent aponeurosis, your fractional anisotropy calculation is instantly corrupted. The numbers will tell you the muscle is failing, when in reality, your algorithm just drew a sloppy boundary.
So the challenge here is that adjacent thigh muscles share nearly identical pixel intensities on an MRI.
Yes, standard edge detection algorithms, even the really sophisticated ones, they look for sharp gradients and contrast to draw a boundary. But the fascial planes separating these muscles do not provide that contrast. It's just a continuous, murky gray landscape.
So to counteract this, the researchers utilize high-resolution 3T MRIs, specifically employing ideal water-only sequences as nnU's DTI sequences.
IDEAL stands for iterative decomposition of water and fat with echo asymmetry and least squares estimation. It separates the magnetic resonance signals of water and fat. By looking exclusively at the water-only images, the researchers stripped away the bright signal of subcutaneous and intramuscular fat.
So they basically optimize the hardware physics to get the cleanest possible image first.
Exactly. But even with these advanced sequences, automated algorithms still fail to separate the individual muscles reliably. The contrast just isn't there to draw a clean line.
Historically, the only solution has been a trained individual manually drawing a boundary around all 14 thigh muscles through the entire 3D volume of the leg. It is incredibly prone to human fatigue, subject to inter-rater variability, and it makes large-scale longitudinal studies financially and temporally impossible.
Nobody has the time or the grant money for that. So, recognizing this bottleneck, the researchers had a human expert manually annotate just 9 key slices for each muscle.
Only 9 slices out of the entire volume?
That's right. They then fed those 9 anchor slices into an automated 3D propagation algorithm utilizing diffeomorphic registration to spread the segmentation along the length of the thigh.
And once the algorithm propagated those boundaries, the researchers went back in and performed minor manual corrections to the edges.
Yes, they termed these assisted manual segmentations, or seg-M. However, this semi-automatic method is a way to build a training dataset, but it is not a viable clinical tool. It's still a bottleneck.
Right. Deep learning promises to take that annotation burden down to zero. Feed the raw scan into a train network and it outputs the volumetric boundaries and the qMRI biomarkers in seconds.
Which transitions us directly into the core experiment of the paper, where they set out to find the minimum effective dose of data.
If you're developing a new pharmaceutical, you run a dose-response curve, right? You want to find the exact milligram concentration that yields maximum clinical benefit before you hit diminishing returns.
And here, the researchers treated the number of manually annotated patient scans as the drug. They took their pristine assisted manual segmentations and fed them into an open-source deep learning framework known as nnU-Net.
How did they structure the experiment?
They structured the training in incremental blocks. They trained a model on just 5 subjects, then 10, then 20, 30, 50, all the way up to a maximum of 120 subjects.
And each iteration was tested against a fixed, totally independent test set of 41 subjects.
Exactly. That independence is key to a fair comparison.
This is how you prevent a network from memorizing the training data. By holding out that independent test set, they could watch the model's true accuracy evolve as they increase the data dosage.
And to measure that success, the researchers utilized a rigorous grading scale. They didn't just rely on the standard dice similarity coefficient, because the dice score is kind of a blunt instrument.
What makes it a blunt instrument?
It measures the general volumetric overlap between the AI's 3D shape and the human ground truth. But a model can achieve a high dice score while still generating jagged, anatomically impossible boundaries that completely ruin a biomarker calculation.
So they layered on strict geometric metrics instead.
They did. They tracked the relative volume difference, or RVD, to ensure the total calculated muscle mass remained within clinical tolerance. They also evaluated the average symmetric surface distance, or ASSD, which computes the average distance in millimeters between the surface mesh of the AI's model and the human model.
And the toughest metric was something called HD95. Think of it as a penalty for your single worst mistake. It finds the one spot where the AI's boundary deviates the most from the true boundary.
Right, so if an AI model is tracking a muscle boundary beautifully for 20 slices, but then on the 21st slice it suddenly spikes outward and includes a chunk of subcutaneous fat, the HD95 will flag that deviation immediately.
And in the context of quantitative MRI, that localized spike is catastrophic. That chunk of fat will aggressively skew the fat fraction biomarker for the entire muscle.
Which means your data is useless. So the researchers set really aggressive clinical thresholds. The model needed an RVD below 5%, an ASSD below 1 millimeter, and an HD95 below 4 millimeters to be considered viable.
When they trained the nnU-Net model on just 5 subjects, the model immediately achieved the dice score of over 0.90 across all 14 muscles.
But the stricter geometric metrics revealed the truth.
Exactly. At 5 subjects, the boundaries were still too volatile. The HD95 and the volume differences were outside acceptable clinical parameters.
But the critical inflection point, the minimum effective dose where the geometry finally crystallized, was exactly 20 subjects. The relative volume difference dropped below 5%, and the HD95 fell below 4 millimeters.
Let's theorize why 20 is the magic number here. If you consider the variance of human thigh anatomy within a controlled cohort of professional football athletes and patients with acute ACL tears, the morphological permutations are not infinite.
That's a great way to put it. After 20 subjects, the network has been exposed to the standard spectrum of healthy volume and the typical geographic patterns of post-injury atrophy. It simply does not need 10,000 examples to infer the geometric rules of a vastus medialis.
And high-fidelity data captures that really efficiently.
But verifying the physical boundaries is only half the battle. The ultimate validation relies on the biomarkers. Does the AI-generated contour actually yield the correct physiological data?
How did they actually test that?
They superimposed the automated segmentations onto quantitative maps and compared the resulting data to the human-drawn ground truth.
And the results were effectively indistinguishable. The interclass correlation coefficients, or ICCs, for the fat fraction and the DTI metrics were greater than 0.98.
Which is basically perfect. An ICC of 1.0 represents perfect absolute agreement. So at 0.98, the data from this automated AI is statistically identical to what you'd get if an expert radiologist spent hours agonizing over every slice by hand.
And to contextualize this, the researchers benchmarked their 20-subject model against a generalized open-source model known as MuscleMap.
MuscleMap is a large-scale algorithm trained on vast, highly varied datasets. And on this specific independent test set, MuscleMap achieved a dice score of just 0.65. On the other hand, this paper's model, trained on just 20 local patients, hit 0.94. The old adage of garbage in has a corollary: perfection in, and you only need a drop of it to teach the machine.
And the researchers have democratized this entire workflow, right?
Absolutely. They've made their 3D propagation tool, named SegPropa, and their final trained model, SegAI-Thigh, entirely open source and freely available to the research community.
We're coming to a close of today's Radiology Advances podcast. Thanks to Slutsky-Ganesh and colleagues for their work published in Radiology Advances. If you found this episode informative or have any other feedback, please rate, review, and subscribe.
You can read the full article and explore more great content on the journal website. This episode was generated with the help of AI, but all content was reviewed and verified by editor Dr. Nelson Gill from Massachusetts General Hospital for accuracy. Thanks for joining us.